So that should be all right. That should be important. So first thing I want to start with is a bit of a recap over what we did last time, which was buffers and carbohydrates. Buffers, I think, are okay. And if you want, you can go over the last lecture to see if there's anything that stands in your way or anything. You can have a look at the last lecture. I send the link on the resources group. And for those who are new to the resources group, we can send everything again. Or if you want, you can message us privately, me and Haitham. So, carbohydrates. What we talked about, what makes a carbohydrate a carbohydrate? Two things, having a carbonyl group and having two hydroxyls. Carbonyl, there are two types of carbonyls, and all that means is that it has this C double bond O, a carbon double bonded to an oxygen. And the other requirement to be a carbohydrate, which we already talked about, is having two hydroxyl groups or alcohol groups. And if we want to know what that means, we can go here. This having an OH, anything that has an OH, you can see an OH in its formula, that means it's an alcohol or has a hydroxyl group. So if you have two hydroxyl groups and you have one carbonyl, that means that, congratulations, you are now a sugar. So these, in the picture below, we can see types of sugars that are important. We've already been through that. Classification of sugars. I'm saying this stuff because I saw that it was repeated in the other lecture as well. So that should be okay. All this stuff you guys can go over. You can go over isomerism. Again, I've already mentioned that, and we don't have enough time. So if you want, you can go back to the first lecture. I can send you the link, and everything is explained in I think good detail. So first thing I want to try, I want to start with today is glycoproteins or N-glycosides. So first of all, we were talking about sugars having a carbonyl group and alcohol group. So what can happen is the carbonyl group, which we can see here, I'm pointing at it. Can you guys see my cursor? I don't know. It can react with the amine group in the amino acids of proteins. Okay, so first of all, what's an amine group? What's an amino acid? If we go back to here, an amine group is anything that has nitrogen and two hydrogens. Okay, and these are called amines. These tend to react with quite a lot of stuff, but in this case, they're reacting with an aldehyde in the sugar. Okay, or in this case, glucose. So glucose is an aldehyde, has carbon double bond oxygen and the hydrogen. Okay, so this amine or amine, whatever you call it, what it does is it reacts with the aldehyde. So the oxygen that has a double bond basically gives off one bond to the nitrogen, making an amide bond. And you can see that over here. Here.
I have a question.
Yes, of course.
What's the difference between a carbonyl group and an aldehyde group? Is it any different? Or?
Well, an aldehyde is a carbonyl. A carbonyl is anything that has carbon, double bond, oxygen. So it may be the difference. Why we call it aldehyde is because it's in the middle. Okay, so it's in the middle of a chain. Let's say like this. This is a difference between. No? Okay. Okay. So yeah, like I said, an aldehyde is basically, sorry, at the end of the chain, if you have a carbonyl, carbon, and then double bond to oxygen, at the end of the chain, then it's called an aldehyde. If it's in the middle, so you have whatever, you have a chain to the left and you have a chain to the right, and the carbonyl is in the middle, like here, you can see here, it's called a ketone. Okay, and that's exactly what makes the difference between aldoses, which are aldehyde sugars, and ketoses, which are ketone sugars. Okay, because some of you are new today, like some stuff is going to sound strange or foreign, if it's something that I have been over, you'll find it in the first lecture. Okay, so now we can form a bond between a protein or an amino acid and the sugar. So we make glycoproteins, which are a type of oligosaccharides, meaning that you have a sugar and you have something else, in this case, a protein. Okay, an example of that would be glycated hemoglobin, or you have here N-linked, what's that say? N-linked glycated hemoglobin. Basically, it's like you can see the structure here. The nitrogen in the amine or the amino acid has linked to the first carbon of the chain in the sugar, okay? Making a glycoprotein or an N-glycoside. And the bond between this bond here or that bond here between the amino acid or protein and the sugar is called an N, because there's a nitrogen, and for nitrogen, glycosidic bond. Okay, so why this is important is because this is the basic structure of nucleosides, okay, which make deoxyribonucleic acid, DNA, okay? So these will form later parts of DNA. So polysaccharides. We talked about polysaccharides as being polymers, okay? They contain multiple repeating units of something, okay? Polysaccharides, saccharides meaning sugar, so polymer of a sugar, okay? And there's two types. There's a homoglycan and heteroglycan. Homoglycan is when it has the same type of repeating unit over and over again. So in an example, we talk about amylose. Amylose is a chain of glucose. So glucose linked to glucose, linked to glucose, and so on and so on and so on. So yeah, there are two types of in this case a chain of glucose. There can be a straight chain and a branched chain. So amylose is a straight chain. So what happens is you can see here what I'm pointing at is amylopectin, but this part is straight. Okay, so the first carbon is linked to 1, 2, 3, 4 to the fourth carbon, and then the first carbon is linked to the fourth, and then first, fourth, and so on. So it doesn't branch, it just makes a straight chain of glucose, okay? And each monomer is linked to the next monomer by glycosidic bonds, which we talked about in the last meeting. However, you can also get branched chains of polysaccharides. And an example of that would be amylopectin. So first of all, what sense does it make when it's branched? Why does it have to be branched? Because when it's branched, first of all, the link between the branch and, so let's say, just a second. Yeah. So let's say here we have a chain, a straight chain, the straight chain part of the amylopectin, and we have the branch here, which is carbon 1. This is carbon 1 here, carbon 6. Okay, so when you cut off that link, that glycosidic bond between carbon 1 and carbon 6, what you will get is you'll get a free chain, which you can then use for ATP production. You can break that down into certain glucoses, and then it goes into aerobic respiration. So that's one thing. That's one thing you get out of chained sequences of glucose. But also it makes it more compact, so you can store more of it because it's branched. It's like you can make it more into sort of fit it into less surface area. Okay, so a good example of homoglycans are amylose and amylopectin, and also glycogen. Glycogen has a very similar structure to amylopectin, but the only difference is it has more branches. So it has 1 to 6 bonds. Carbon 1 to carbon 6 here. Carbon 6, carbon 1 are linked. Okay, so when it has more of that, meaning it has more branches, meaning it can be more compact, meaning you can break off more chains and so on, use them for energy, which is why glycogen is an amazing energy store found in the liver, of course. And another example of that would be cellulose. Cellulose is similar to the concept of glycogen, but it's made out of beta glucose. And we did go over the difference between alpha and beta glucose in the first lecture, if you guys want to check that out. So yeah, the only difference between beta glucose and alpha glucose is that beta glucose does not coil around itself, just as straight as it comes. Okay. And you have also chitin. Chitin is a polymer of N-acetyl glucosamine. We don't need to know what that looks like for now, but it's used the same way as cellulose is, but for insects. So insects have chitin, which is a polymer of N-acetyl glucosamine, and they use it to protect themselves. So that makes up the exoskeleton. When you crush an insect, and it kind of, I don't want to make it disgusting, but it kind of crunches, you know. That's what you're crushing, is the exoskeleton made out of chitin. Okay, so now we move on to heteroglycans. This is where it gets tricky. Heteroglycans are made out of more than one type of monomer, okay? Or in this case, a sugar, okay? So let's say we have a look at heparin. Heparin is, by the way, it's a polysaccharide used in this immune response, okay? So here you can see the structure of heparin. It has two types, okay? It has D-glucuronate 2-sulfate. And what all that means is that it has a sulfate here on the second carbon. Okay, it's a glucose and it has a sulfate on the second carbon. And it also has a carboxylic acid group on the sixth carbon. So this is 1, 2, 3, 4, 5, 6. We're there. Okay, and it's linked to another modified glucose, but these two are different. They're different monomers. And what happens is when you put these two together, you will have them in repeating units. So you'll have that, and next to it the same, and next to that the same, and so on and so on. Okay, so that's why it's called a heteroglycan, because it has two different types or two different structures of monomers. Okay, and heparin also mediates the immune response. So when you have an allergy or something like that, you'll find heparin, and also acts as an inhibitor of blood clotting cascade. So there is something called the blood clotting cascade, and it basically, the end result of it is you will get a blood clot made out of fibrinogen. And heparin, what heparin does is that it stops this cycle, this cascade. Okay, and so it's used in anti-clotting drugs when someone has their blood clots a lot and therefore they're a risk of stroke or heart attack. You give them heparin, among other stuff. Another example of a heteroglycan is hyaluronic acid. Okay, so hyaluronic acid also, as you can see here, is a heteroglycan because it's made out of two types of monomers. Okay, this is in the middle, the thing that I'm pointing at here. This is the glycosidic bond. Okay, and on the right you have this structure, which I think is amine 2 glucosamine, and you have this structure here, which is glucuronate, because you have carboxylic acid group here. So anyways, you have these two different monomers. So before it's a heteroglycan, okay? And they repeat to make up the polymer of hyaluronic acid. Hyaluronic acid is very important, and we use it for a bunch of stuff, and it also makes up other molecules. One thing it's used for is that it binds water. And why this happens is because these bonds, the carboxylic acid group especially, is very soluble in water. Okay, so you can dissolve water in this, and water can be bound to that molecule. Okay, and as you can see here, one molecule of hyaluronic acid can bind up to 250 molecules of water, so that's quite a lot. And these, because they have an amine group, okay, and they are sugars, they are kind of oligosaccharides. So this part of the hyaluronic acid, and also you can find it in heparin. They are called glycoaminoglycans, amino, meaning that it has a nitrogen in it or an amino group. Okay, same as you can see here, and as you can see in the first heteroglycan, heparin. Okay, so we'll move on for now. Lipids. Okay, so now that we're done with the nightmare that is heteroglycans, we'll go on to lipids. Not that it's easier. So lipids. Lipids, you have different kinds of lipids. I don't like this structure very much. Let me find some. I think this is the presentation the teacher used, by the way. There we go. So I don't like this either, but it is kind of better. So first of all, lipids, there's simple lipids, complex lipids, and lipid-like molecules. So these are not lipids, but they have similar properties sometimes, and they include steroids, and I don't know how to pronounce this, but I think it's science. I don't know, cyanoids. But anyways, the most important lipid that we're going to talk about now, or one of the most important lipids, is triglycerides. You can see here, triglycerides is your very basic, very standard fat. Okay, so they make up most of the adipocytes, which are fat cells. Okay, and therefore they make adipose tissue, fat that you can see in our body. Okay, and this has a lot of uses, like for whales, for example, it keeps them afloat. For humans, it keeps us warm, acts as energy storage, a lot of stuff. But yeah, triglycerides. Now let's talk about the chemical structure of triglycerides. Triglycerides are made out of two types of molecules: glycerol, alcohol. So it has three hydroxyl groups. Okay, OH here, OH here, and OH here. And the other type of molecule is fatty acids. So fatty acids are, like we said, acids, so carboxylic acids. Okay, you can see the carboxylic acid group here. But if we want, we can refer back to the functional groups. Here you can see carboxylic acid, C double bond O, and then O double single bond H. Okay, so that's a carboxylic acid, and the other one is an alcohol. What isn't here though is that alcohols react with carboxylic acids to form ester bonds. Okay, and that's exactly what you can see here. This here is called an ester bond. O and then C and then double bond O. You can even refer back to the functional group chart and see esters, C double bond O, and then you've got an O here. Sorry. Just making sure that everyone's inside. Okay. So now that we've gone through esters, you can see the full structure of the triglyceride, which is made out of glycerol, one glycerol molecule attached to three fatty acid molecules, making kind of this shape. Okay, and this then forms your standard fat or adipose tissue. But also it forms waxes. So waxes are material made out of triglycerides, and what they do is they repel water. So one thing we need to learn about lipids is that they repel water. Okay, and of course some of them repel it partly, some repel it completely. Triglycerides repel water completely. They're very water phobic, water afraid. So they don't like water. They repel water. Okay, and that's why they make waxes. Okay, so waxes are found on feathers, and fur, on the skin, because it repels water. So you don't soak in water, and it doesn't clog your pores and so on. And of course, if you're a bird and it's quite cold outside like it is now, then that's very bad news for you. So that's why you have waxes, which are made out of triglyceride. Okay, so the next lipid molecule that we're going to move on to is phospholipids. Sorry, I put the message in it. Is it enough for the exam just to read the presentations? Ooh. If you understand it, then it should be enough. Like, let's say, if I read the presentation on triglycerides, you know, a lot of the questions on the exam sometimes would be application. Okay, so you need to apply your knowledge. So you need to understand. Okay, so you don't really need to. Well, some stuff you need to know by heart. You just need to memorize it. But most of the time, you will be okay understanding what's happening. Okay, or read Harbour Biochemistry. Well, the chemistry book that I used to get through chemistry last year was the Handbook of Chemistry, the University Handbook of Chemistry, and you can find that from the second years, or you can ask the university about it. And yes, that's all I needed, to be honest. And a lot of the questions of the exams, I could find the answer to from the handbook. But obviously, any book that you have or any website is going to be very helpful, okay, or helpful to some extent, you know. So yeah, YouTube is also very, very good in explaining stuff. But the problem
I have a question.
Yes, go on.
No, just say your problem first. I'm sorry.
No, it's okay. I'll come back to that. You can say your question.
Yeah, so my question was that the teacher recommended to use the book Timberlake if you weren't very good at chemistry.
Yeah.
Do you recommend that one for the people
Honestly,
that are not?
I can't recommend it because I haven't tried it, to be honest. But I'm glad to have a look. I can have a look at it and tell you whether it's going to be useful or not. Because the thing is, if you're not very good at chemistry, it's all about foundation. Okay, all you need to understand what's happening in most of the chemistry syllabus is having enough foundation. Okay, so if someone doesn't know what a lipid is, okay, and you're going on like the teacher says, or you're skipping over lipids, or you're skipping over some basic stuff that the teacher thinks is basic, but some people actually don't know. And then you talk about sphingolipids. I'm not going to understand if you talk straight away about that. So it's all about foundation. If the book can give you the foundation that some people don't have in this part, then it would be helpful. If not, then it's going to be hard to understand. So it's all about taking the first steps. As long as you get the first steps out of those books, and like, what is a lipid? Okay, what are the types of lipids? What's an alcohol? What's a carboxylic acid? What's an ester? All that stuff. So if it has that, is going to be helpful, I think.
Okay, and my second question was, are there any like formulas or anything that you think we should memorize by heart?
I'm not a fan of memorizing formulas, but there are some that I think you're going to need. So first of all, for the calculations part, any formula that includes number of moles, concentration, how to convert between units. But these are, you see, if you want to memorize a formula, all you have to do is understand it. Okay, if you understand the formula, how it works, what it does. So let's say if it wants me to convert between percentage to millimole per liter, well, percentage, I need to know what percentage means. Percentage means milligrams per 100 milliliter. Okay, so I can say, okay, that's fine, and then it will give you the molecular mass of a certain substance, and then you can find the concentration of it. So once you find the concentration in moles per liter, then you can find the concentration in millimoles per liter. So in that sense, there is an equation, but if you understand, you won't have to learn the equation itself. But there are some stuff that you're going to have to learn by heart, which is N equals C times V. which number of moles equals concentration times volume in liters. And yeah, there are some formulas, and maybe in the future I can put them in one place to help people memorize them. But yeah, I think hopefully that should answer your question.
Yeah, it did. I hope that you're going to go through the something Hasselbalch equilibrium or something like that later.
Yeah,
because
no problem.
I tried to understand it and I got halfway and then I fell off.
Have you checked the first meeting, the recording for the first meeting?
Yes, I did.
No problem. When you have time, you can do it. And then maybe if that doesn't help, then you can tell me.
Okay, thanks.
Problem. Okay, so back to the topic. I think there was a question I was answering. Yeah, no, that's okay. Back to the topic. So phospholipids. Phospholipids are another type of lipid. And from their name, we can say that they have phosphate group. Okay, here we can see a phospholipid, and you can see that it has a glycerol, which glycerol alcohol, having two fatty acid chains. By the way, oh yeah, I didn't go over something important. Fatty acids, there are two types of fatty acids: saturated fatty acids and unsaturated fatty acids. So saturated fatty acids have a long chain. There are fatty acids, normal fatty acids, long chain of carbon and carboxylic acid group at the end, and they don't have any double bonds. So they don't have any C double bond C. Whereas unsaturated fatty acids have two or more double bonds. Okay, so you'll find a chain and then you'll find a double bond like so, like this, exactly like that. And then, yeah, if it has more than one double bond, then it's called polyunsaturated fatty acid. So phospholipids, you have one saturated fatty acid and another unsaturated fatty acid. Both of them bound but to the glycerol by ester bond, which we already went through. Alcohol, carboxylic acid makes ester. Okay. But here's the trick. With phospholipids, you also have a phosphate group. So a phosphate group will look something like this. Like so. Okay. So here, the R, meaning that there's a chain, or yeah, there's a chain here. Okay. And you'll have these hydroxyl group, and you have an oxygen. You have an oxygen here as well. There's double bond oxygen. So all of these can react. And we talked about, just a second, the double bond in the oxygen can react with the alcohol. In the carboxylic acid, sorry, can react with the alcohol. So it works the same way for the phosphate. You can see here, sorry, one of the OHs, so the hydroxyl group, reacted with the glycerol. Okay, so water is condensated after that reaction, because here you can see the phosphate has lost the hydrogen and the glycerol has lost the hydrogen. Okay, so it's a condensation reaction, and so on. You get the phosphate is bound to the glycerol. Okay, but wait a second here. All of this is insoluble in water because the oxygen is very deep inside the long chain. Okay, but the phosphate, you have an O minus here. This is polar. Okay, and it's very exposed. So this part is soluble in water. So you get a hydrophilic, a part that actually dissolves in water, and a hydrophobic part, which doesn't dissolve in water, which we talked about before, which is the fatty acid part. Okay, so you essentially get kind of a head that can dissolve in water and a tail that doesn't. So when you have this formation, what will happen is the part that's non-soluble in water will be like, well, I want to stay away from this part as much as possible. So when you put two of these together, you will get that shape, the bilayer, phospholipid bilayer. The part that doesn't dissolve in water, that dissolves in lipids, will be like, okay, I'm going to dissolve in other lipids. So that's other fatty acids, other non-water soluble parts. So you get the hydrophobic tails are against each other like this, and the hydrophilic heads are away. So there's the hydrophilic head, there's the hydrophobic tail, all of that. Okay. So on the hydrophilic part, you can get water soluble molecules. Okay. But on the hydrophobic part, you can get lipid molecules or lipid soluble molecules like cholesterol. Okay, which we'll go through later. So you will actually find cholesterol within the lipid bilayer because it's lipid soluble. Okay, so now we'll go over phosphoglycerides. Phosphoglyceride, this sounds very complicated, but essentially all it is the phospholipid. It's basically a type of phospholipid, but you see this R group, okay? It can be anything, okay? So it can be, hold on just a second. So this, yeah, so this part acts similar to carboxylic acid, and therefore you can form esters out of it because carboxylic acid plus alcohol is ester. So anything with a hydroxyl at the end of it or an alcohol group at the end of it, you like ethanol amine. You can see here the hydroxyl group OH or HO, serine HO, and also carboxylic acid group. Choline is not there, but it does have an OH. Incitosol, all has 6 OHs. Okay, so all these have OH at the end of them, and therefore it can react with the phosphate group to form phosphatic esters. Okay, which are phosphatic acid esters, which are phosphoglycerides. Okay, and some of these include. Hold on, is it not in here? I might have missed it now. That okay. So hold on, let me try and find it. There we go. Second presentation. Thanks to whoever gave me this presentation, by the way, it's very useful. There we go. So examples of phosphoglycerides are phosphatidylserine, which is, as you can see here, you have the phosphate group, okay, and you have the glycerol, okay, and you have the 2 fatty acids, okay, and you also have a serine, okay, which is an amino acid. So yeah, you can have all kinds of reactions because that phosphate here, the phosphate at the end of the phosphoglyceride has a lot of functional groups that can react with other stuff, okay? Partly forming phosphoglycerides, which are phosphatic acid esters. So alcohol, carboxylic acid, ester. Okay, now we move on to plasmalogens. So what are plasmalogens? First of all, I noticed that you guys are awfully quiet today. If I'm going too fast or too slow, or there's something that I skipped over and you guys want to ask any questions about it, then please do so. I don't want people here to be sitting and not understanding, because all I'm trying to do is I'm trying to fill in the gaps. I'm afraid the gaps here are very big and very hard to fill, but I'm trying. So if you guys have any questions, or there's something that you don't understand, or a word that you don't know what it means, okay, then it's okay to ask. Okay, that's what we're here for. We're here to learn. So if you guys have any questions, any words that you don't understand the meanings to, then please don't be afraid to ask. Okay.
I was thinking about if I can also make like a sort of meeting where I can explain ester and all the groups, because I feel like that's where I think most people get confused with.
Yeah, sure, no problem. Well,
Yes.
Yeah, go on. Sorry.
No, I only said yes to the thing Maisel said.
Okay, no
Yeah,
problem.
I feel like this is like if you don't know the groups, I guess it's going to be hard to understand the rest, I guess. Yeah.
Okay, so I'm happy to do a meeting on whatever you guys struggle with, of course. But I have a solution. So if you don't know what the functional groups mean, first of all, there's this chart. It's not much, but it is something to go off. And also, you can, like this stuff, you learn by experience or by practice. Okay, so glycerol, I know straight away that's an alcohol because there's an O in it. Okay, a fatty acid, I know that's a carboxylic. I know that's a carboxylic acid because there's acid in it. Okay, so you can look everything up, of course. So if I don't understand what something means, as long as I have this chart, then I can say, or I don't know what a reaction is, I can say, okay, so I don't know what this reaction produces or something that I can go, for example, here, the phosphoglycerides, for example. I don't know how that works. Okay, so I'll go on Google and say here, phosphoglyceride. Okay, and then you can see, trying to find the reaction. Even. There we go. That's better. So now I can refer back to the chart. Okay, here we have ethanol amine, which has an OH and an amine. Okay, and we have choline, which has an OH and an N plus. Okay, so now if we mix these two, okay, or if we mix them with a phosphatic acid ester, or sorry, phosphatic acid, so we can make a phosphatic acid ester. So here you can see the phosphate, and it reacts with which part? The alcohol part, OH. I know what's OH. You can go back to the chart and say, what's OH again? What's OH? Oh yeah, it's an alcohol. So I know this is a reaction between an alcohol and a phosphate. Okay, so that's it. If that helps, then by all means use the chart, and you can go on Google and of course Google anything that you don't understand. But if there's still a lot of people that struggle with it, then we can have a meeting over that. I don't have a problem with that. Actually explains a lot. And YouTube, yeah, for that stuff, it's like this is the stuff that I was talking about earlier. These are the sort of building steps for everything that you're going to learn later. Okay, you can learn them from YouTube, or even if some of you did A levels or high school stuff, could be there. Okay, but not necessarily. So yeah, we can make a meeting over it, definitely. Okay, anyways, back to the lipids, to the plasmalogens. So plasmalogens are a type of glycerophospholipids. So these are the ones that were the phospholipids that we were talking about, glycerol and phosphate and 2 fatty acids. Okay, which contain an ether and an ester bond. Okay, at an SN1 and SN2 position. So all that means is, even though it seems complicated, it really isn't. So you have here, this is the glycerol molecule, everyone. Okay, and this is the SN1. Hold on. This is SN1, and this is SN2. Okay, and at each one of them, respectively, you have an ester. What's an ester? For those people who are saying, I don't know the functional groups, can we do a meeting over that? What's an ester? I can go back to the chart here and say ester is C double bond O, and then you have an oxygen. Okay, so yeah, I'll go here and I'll say, okay, so oh look, that's an ester. Okay, what about an ether? We have an ether here. Okay, so we'll go down. What's an ether? Ether. Yeah, there we go. Something, and then oxygen in the middle, and something else, which we can see over here. Carbon, oxygen, carbon. Okay, so if they have an ether and an ester, okay, and they're in a glycerol molecule, then it's a plasmalogen. Okay, so the groups bound to the glycerol an example of something that can bind to the glycerol here or here is choline, alcohol. Okay, so it can form an ether, or ethanol amine, ethanol, alcohol hydroxyl group can also react, and so on. So this is an example of a plasmalogen. You can see here the phosphate group. Okay, and it has an ether bond. You can see there's a circle in it, and an ester bond right over it. Okay, so this is a plasmalogen. So what are plasmalogens used for? Plasmalogens are platelet activating factors. Okay, so what that means is they play a role in blood clotting. Okay. Yeah, so for example, during the development, okay, you can find plasmalogen a lot because it sort of forms platelets or activates the platelets so that some parts of the organs can form. You don't need to know much about that, but all you need to know about plasmalogens is that they activate platelets. Platelets are substances found in the blood, and they play a role in blood clotting. So, hope we can move on. Sphingolipids. So sphingolipids are made out of sphingosine and fatty acids. Now that doesn't make sense because we don't know what sphingosine is. But literally all it is that it's a carbon chain, and it has double bond alcohol, an amine, and another alcohol at the end of the chain. Okay, so you don't need to know what sphingosine is made out of. It's made out of, I think, coenzyme A and something else. But anyways, sphingolipids are basically this molecule, this sphingosine molecule, whatever it is. As you can see the structure here, okay, attached to a fatty acid. Okay, so here we have an amine group, the one in the circle. Okay, this amine group can react with carboxylic acid. Okay, so what's the carboxylic acid again? Down. Here we have amine, so NH2, and carboxylic acid. And what that makes is when these two react, they make an amide link. Okay, forming sphingolipids. So fatty acid plus the molecule called sphingosine. Well, I really don't know how to pronounce it. Makes sphingolipids. And yeah, like I said here, an amide bond. This is an amide bond, the one between the amine and, well, you can't see it here, but it's the carboxylic acid making an amide bond. Let me see if I can find a picture of an amide. There we go. Looks something exactly like this. You see the one in red? That's the amide bond. So carbon double bond to oxygen, and also double bond, so single bonded to nitrogen. So that's an amide bond. Okay. So yeah, sphingosine, which contains an amine group, reacts with a fatty acid, which contains a carboxylic acid group, to form an amide link, which then forms sphingosine. Okay. Sphingosines are signaling molecules, and they signal different processes like phagocytosis or apoptosis or synthesis. So this is the stopping of cell growth or differentiation. Okay, So it's a signaling molecule. So like we said, sphingosine, as we can see, sphingolipids, as we can see here, also has an alcohol group, an OH group. And we were talking about, okay, alcohol group can react with so much stuff, but we talked about it reacting with a phosphate before. We were talking about phospholipids and phosphoglycerides, okay? You can see here, sorry. Yeah, oxygen can react with, the phosphate can react with the alcohol group, okay, and form a link, an ether in this case. So an ester link in this case. So here we have the sphingolipid, okay, and the sphingolipid also has an alcohol group in it. So the alcohol reacts with an organic phosphate, and it forms something called phosphoceramide. Okay, so this sphingolipid reacting with phosphate forms phosphoceramides. Amide, ceramide. What's an amide again? Go back to that chart. Amide is this. So carbon double bond to oxygen, and then you have a sort of amine at the end of it. So there we go, phosphoceramide. Well, because the phosphate is so reactive, it can also react again and form esterified phosphoceramides. Okay, so you have one of these alcohols can react with another alcohol, form an ether bond, okay, or it can react with a carboxylic acid or whatever, okay. But the most common stuff that it reacts with is serine or choline or ethanolamine, inositol to make, or inositol, okay, which are all alcohols. They have hydroxyl groups, which are OH groups at the end of them. They can react with the phosphate to form esterified phosphoceramides, okay? And these also have other biological functions. I think we're almost at the 1 hour mark. So yeah, we're not even, but we are close to finishing, but not as close as I'd like. I can sort of see why the chemistry teacher skips over a lot of stuff, because there is a lot of content. It's kind of stupid to have this much content, to be honest. And especially when some of the foundation isn't offered at all high school levels. It depends what country you're from. So yeah, agreed. Thanks, Patrick. Now we'll go over cerebrosides. Okay, so now we have sphingolipids. Okay, what can happen with sphingolipids, because they have an alcohol at the end of them, is they can react with a lot of stuff. Okay, but now we're going to go over the reaction. So sphingolipids, which have an alcohol at the end of them. Here, just a second. Sphingolipid. Alcohol at the end, CH2OH, hydroxyl alcohol, can react with sugars and especially glucose. Okay, so we call this reaction glycosylation. Glycosylation. That's a misspelling, guys. Okay, there we go. Glycosylation, because you're adding glucose. So sphingolipids, the alcohol group in the sphingolipids, can react with one of the alcohol groups. In this case, it is on carbon 6, 5, 4, 3, 2, 1. On carbon 1, so we can go back to the structure of glucose. Just a second. Yeah, you can see on carbon 1, there's an OH group. So alcohol plus alcohol, that's an ether. Okay, which is, if you don't know what it is, we can go to the functional group chart and check it out. So you're adding a glucose to the sphingolipids, and that forms cerebrosides. Okay, another organic substance. Okay, so at the first carbon of the sugar, the OH group reacts with the OH group from the sphingolipids, okay, originally from the sphingosine, okay, to form cerebrosides, okay. I have no idea what cerebrosides are used for, actually. So glucose cerebrosides, the latter is often used in non-neural tissue, so in your skin. And it plays part of water permeability barrier function. I can actually see why that's the case, because it's partly soluble in water. You have here the long chain of the sphingosine. It doesn't have any polar parts, no OH, nothing. So it's non-soluble in water, but this part is soluble in water. So I can see why it forms a water barrier, okay, because it has a non-soluble part and a soluble part. And now we move on to gangliosides, okay? So gangliosides are very similar to cerebrosides, but the only difference is that instead of a glucose, okay, or a sugar, you have an oligosaccharide. Oligosaccharide, again, for those who don't know, in the first lecture, I mentioned that oligosaccharides are sugars with other functional groups. So let's say a sugar and a protein, or a sugar and a lipid, glycolipid, glycoprotein. These are all oligosaccharides, okay? So here, this is an example of gangliosides, and you can see here, you have the N-acetylhumeric acid or whatever, and you have a glucose and you have a galactose, and you also, of course, you have the sphingolipids, okay? Oh, actually, hold on, I don't know if you have this. Is extended to oligosaccharide. Yeah, so you have the two carbon chains, and you have also the oligosaccharide part. Okay. So now we move on to lipid-like compounds. So compounds that have similar properties to lipids, but aren't actually lipids. So for this part, for the eicosanoids, there isn't much to know. You just have to memorize this, to be honest, because I couldn't, like, it's made out of something called arachidonic acid. Okay, and yeah, they're derivatives of a carboxylic acid called arachidonic acid. Okay, you can see it's a carboxylic acid here. It has 1, 2, 3, 4 double bonds. Okay, so which means it's quite reactive, because these bonds can get broken, they can get reformed in other carbons. Okay, the carboxylic acid group can change as well. So these are quite reactive, and a derivative of them is eicosanoids, eicosanoids. Okay, and their uses include hormones, so they can act as signaling molecules. Okay, yeah, so they act as signaling molecules in different processes. For example, the inflammatory reactions, so the release of histamine and stuff like that, it may act as a signaling molecule, so the cell releases other effectors, or histamine or so on. And yeah, cholesterol or steroids. Steroid hormones are made out of steroid derivatives. Okay, and they include androgens, corticosteroids, and so on. But for now, they are derivatives of cholesterol. So what is cholesterol? Cholesterol is a lipid-like molecule, but you can see, unlike lipids, it has very non-water soluble parts. This is like lipids. That's why it's like lipids. But unlike lipids, it has a OH, or unlike most lipids. So it's partly soluble in water, okay? Because this OH is polar, and therefore can dissolve in water. And that's also why it's called amphipathic, because it's soluble in lipids and soluble in water. And can release the H+, because it can also act as a proton donor, or an acid, like we talked about in the first lecture. Okay, So cholesterol, there are two types of cholesterol, high density cholesterol and low density cholesterol. In other terms, the good cholesterol and the bad cholesterol. Low density cholesterol, as you can guess, is a bad one because it forms blood clots. Okay, and that's how you get strokes and heart attacks and so on. But like we said, cholesterol has an OH group, which also means that it can react and form esters. So carboxylic acid plus alcohol, that's an ester, okay? And if you do that, then the oxygen or the polar part of the cholesterol becomes sort of taken inside the bond. It's hidden in the bond, like you can see here, okay? And therefore, the molecule becomes non-soluble in water. The whole molecule becomes hydrophobic, okay? Now we move on to cholesterol derivatives, steroid hormones. You guys have been awfully quiet, so yeah, there we go.
Can I just say something about cholesterol?
Yes, go on.
Go through previous slide. So if you guys see that the cholesterol is used to build membranes, just in case you're wondering, and I think it's a very good thing to add to your notes.
Yes.
So cholesterol is used in the membranes together with the phospholipids that Kareem talked about earlier. And just to let you guys know the importance of it in the cell membrane, it's that it controls the fluidity of the
Yes.
cell membrane.
Exactly,
So
and especially at different temperatures. So cholesterol is kind of like a stabilizing agent.
Yeah, so at high temperatures, it stabilizes the membrane and prevents it from shaking a lot because there's a lot of kinetic energy. And in the opposite situation where there's low freezing, let's say when it's cold, the cholesterol is present and it prevents the clustering of the phospholipids. So
Exactly, so the phospholipid molecules cannot get too close or too
Exactly.
far from each other, depending on the temperature of their environment. Yes, that's actually something very important I forgot to mention. Thank you, man. Yeah, so if there are any questions, you can ask, of course, anytime. It has been hours.
Maybe you said it, but
Sorry?
maybe you already said it, but the cholesterol is then functioning like a buffer, kind of, or
Yes. Well, a buffer to the effects of temperature, but
yeah.
it's not a buffer in the sense of acid and base. I said that
No.
it can be an acid because it can donate the H plus, it can donate the proton. So acids are anything that donate protons. So it can donate a proton, and therefore it uh, it can be an acid, it can be acidic. So yes, but only in certain conditions when the pH is very high and it needs to donate that proton. So cholesterol derivatives. So cholesterol derivatives are molecules that are basically variations of cholesterol or cholesterol that has other functional groups added to it. And good examples include bile acids. So that's the bile fluid in your bile ducts from your liver, okay, and bile salts, which are also very important in digestion. You have cortical hormones. Cortical hormones are important in regulating the fight or flight response, and stress also. So if you're very stressed, you'll find a lot of cortical steroids in your blood, and sex hormones. So these include testosterone, progesterone, and so on. And also vitamin D. Vitamin D is actually a derivative of cholesterol, and it is a steroid. So yeah, as you can see here, the basic structure of steroids is present in all cholesterol derivatives. Sorry, of the basic structure of cholesterol is present in all steroid hormones and steroid substances and cholesterol derivatives. So cholic acid and bile salts, you can see here, it's a cholesterol, and it has this carboxylic acid group attached to it. Okay, you can see here cholesterol, but instead of this, it has a carboxylic acid. Bile salts, you can see here, that it has a different, it's the same as cholesterol, but it has a different configuration. It has an amide. This is an amide, everyone, and it has a carboxylic acid as well. So that's why it makes salt, because it can lose the hydrogen and can accept a hydrogen as well, hydrogen ion. And then we have vitamin D, which also has similar structure to cholesterol, but different in the side chain. Okay, so these rings, the cholesterol ring, you can see it in all steroid hormones, okay, including testosterone. So here, steroid hormones, you have 3 types of steroid hormones. Glucocorticoids, so these include cortisol, corticosterone, and so on. And these, they're important in regulation of the stress response, okay? And you also have mineral corticoids, and these, as from their name, they regulate the mineral content of the blood, and so on. And you also have androgens. Androgens are sex hormones, so testosterone, progesterone, and so on. And you can also see in all of them the cholesterol rings, 1, 2, 3, 4, so on, 1, 2, 3, 4, 1, 2, 3, 4. The only difference is something that you might have noticed is that there's a double bond oxygen at the end of it. This is not present in cholesterol. You have a alcohol at the end of cholesterol. So this is the part that I was apologizing about because I didn't get enough time to do it, but I can go over it as quickly as possible from the presentation that has already been given to me. So proteins. Proteins are made out of smaller subunits called amino acids. We have gone over amino acids before, but for those who weren't here, an amino acid has this structure. So it has a carboxylic acid group and it has an amino group. And this R, by the way, not in this case, but in all R's that you're going to see in a structure or formula, refers to a variation, okay, of the chain. So this R can be anything, okay? So here we have, yeah, the amino group and the carboxyl group. The proteins are made out of amino acids or chains of amino acids. It's not quite simple, but there are 2 types of amino acids: essential amino acids and non-essential amino acids. Non-essential is because you don't need to eat them or you don't need to get them from your diet. Your body normally produces them. But essential amino acids, your body doesn't produce them, so you need to get them from your diet. So, yeah, there are 20 amino acids that all proteins are made up of. Of course, different variations, different sequences, but all proteins are made out of a certain sequence of a twin. Like you have 20 amino acids to choose from. Okay. So amino acids form proteins by linking together. So they kind of, the carboxylic acid group of the amino acid reacts with the amino group of the next amino acid. So it's kind of goes like this and so on. Like it's chain here, chain here. It's a long chain of amino acids, okay, linked by peptide bonds. So a peptide bond is the bond between the carboxylic acid group of an amino acid and the amine group of the next amino acid. But well, we know, because we know chemistry, that we can go back to the chart here, and we know peptide bond, carboxylic acid and amine, that forms an amide. Okay, so a peptide bond is a type of amide. So that's how you form the amino acid chain, which is also called peptide. Peptide is made out of amino acids, and you have first of all, let's move on to the classification of amino acids, because amino acids are classified based on whether they are polar. Okay, so like we said, we have the basic structure, the same structure for all amino acids, but there are variations in the R group. So you can see here, this is the R group. Okay, these are all non-polar R groups. They don't have a hydroxyl or a free oxygen, or yeah, apart from tyrosine, but that's because it's a special case, because there's a phenyl in there. Okay, and phenols are very non-reactive in some cases. Okay, so yeah, generally they're non-polar and they're non-soluble in water. okay, and therefore they're hydrophobic, okay. But we also have polar amino acids, which are soluble in water, okay. There was a question asking to label which amino acids are polar and which are non-polar. It doesn't make sense to know them, to be honest, but well, no, I would say know about 4 of each, okay? So out of the non-polar, you can know glycine, alanine, valine, and leucine, okay, or the most basic ones. And for the polar as well, I would know a few, because sometimes you're asked to label amino acids, especially in the protocols. That was one of the protocols, one of the quizzes, I think. So amino acids are amphoteric, okay? They can be acids or they can be bases because they have a carboxylic acid group. And the carboxylic acid group, as we know, can donate a proton, can donate a H plus hydrogen ion. So therefore, it's an acid because it can do that action, okay? But also, it can accept a H plus, it can accept a proton, making it a base, okay? So they can act as either, depending on the pH of their environment, and they're something called isoelectric point. So what an isoelectric point is, it's the point of neutral, okay? So it's like where you can get the carboxylic acid group to donate its proton, but you can also get the amine group to accept a proton, okay? So it gives you the structure that is circled in red, okay? And that's called the Zwitter ion. Might ask you to draw a Zwitter ion, because I think that was one of the questions as well. And the Zwitter ion occurs at the isoelectric point, which is the point where the amino acid has 0 charge, okay? Isoelectric point, by the way, is a pH. So there are some amino acids that are used as amino acids, okay? They're not linked to proteins or anything. They're just used as themselves. And a great example of these is aminobutyric acid, okay? So this is that's used as a neurotransmitter, and it doesn't belong to a protein, okay? Same with aminocapronic acid, which forms an inhibitor in blood clot formation, okay? You also have taurine and citrulline and ornithine, but you wouldn't get a lot of questions about these, but do know them, especially citrulline and ornithine, because they are part of the ammonia cycle. You can Google that in your own time. So amino acids do have an amine group, which means that they can react with carboxylic acids to form amides, okay? So an amino acid such as, or a protein such as histamine can have its amine group react with the carboxylic acid group to form an amide, okay? Or you can get rid of the carboxylic acid group to form just the amine, okay? And you get, if you do that with a histidine, which is a protein, you will get histamine, the very famous signaling molecule that is used in the inflammatory response. And this is very important. Yes, exactly. And if you do that to tryptophan, so if you take the carboxylic acid group out of the tryptophan amino acid, you will get tryptamine, okay? Then tryptamine forms serotonin and melatonin, okay, which are very important also signaling molecules, pleasure and happiness and so on. So like we talked about for peptides, peptide chains are a chain of amino acids, and they are formed by amino acids forming a chain. So the carboxylic acid of a group reacting with the carboxylic acid of a different group. Wow, a lot of people has. I have to go now, but I appreciate you loads. See you soon. Okay, yeah. So I think I'm going to end it here because because I have taken a lot of your time, but it's a very long topic, and it has a lot of stuff that you should cover in weeks, you know, and they're just doing it in one lecture, which doesn't make any sense. But yeah, I tried as much as possible to simplify some of the stuff here. So I hope it helped. Like we said, we can discuss meeting to talk about functional groups and thank you. Yeah. Yeah, we can make a meeting about functional groups and different biological and organic substances and molecules. If you guys want, please message me about it. And if enough people do it, then we'll definitely have one in the near future, hopefully. Okay, thanks, guys. I think that's it. So whenever you're ready, if you don't have any questions, of course, if you have any questions, then feel free to stay behind and ask. So yeah, that's it for today. Well, that was a lot of talking. Thanks, guys. Thank you. So I'm going to end the meeting there. Of course, Haitham, you can stay behind. It's just me and you now.